Thank you. Well, good morning. It's my pleasure to tell you about some of the technologies and development activities at Scancell over the next 15 minutes or so. I'll just bring your attention to our disclaimer notice. I'm sure you can all read that very quickly. Okay, so Scancell's an immuno-oncology company. And I'm sure you're all aware that the immuno-oncology market now is a rapidly growing market, mainly on the back of the success of the checkpoint inhibitors such as Keytruda. And of course, despite the success of these drugs, they are obviously not a panacea in cancer. So there's a huge patient population that don't respond to these therapies. So obviously the race is on now to try and find what are the optimum combinations or other modalities where we can actually address the unmet needs in this non-responder patient population. And this is exactly where Scancell's technologies come in. So we have 3 platform technologies: Immunobody, Moditope, and more recently we announced this new technology called Avidimab. And from those platform technologies, we now have 4 products in development. 2 of those products are actually now in clinical development, or in fact, well, one is in a phase 2 clinical trial now, with the other following very rapidly into the clinic in the first half of next year. And of course, we're addressing multiple cancer indications with these products. The company was founded by a renowned immunologist, Professor Lindy Durrant. We have our head office in Oxford, and the company is AIM listed with the ticker SCLP. And we have a number of partners, including BioNTech and Cancer Research, and I'll talk a little bit about those relationships as we step through this presentation. So this is our development pipeline. So as I mentioned, 3 platform technologies. Immunobody and Moditope are both cancer vaccine platform technologies, and then we have this panel of monoclonal antibodies targeting tumor-associated glycans, and we'll talk a little bit about those as well. So our lead product is an immunobody, which is called SCIB1. That's been through a phase 1/2 clinical trial in late-stage malignant melanoma. 2 cohorts of patients. We've shown efficacy and also safety in that study, and now, as I mentioned, we have initiated a phase 2 combination study here in the UK. SCIB2 is another immunobody that is in collaboration with CRUK. CRUK have taken that product, and they're now orchestrating a clinical study, again a combination study, 1/2 study, which we anticipate will be initiated next year. And then our lead Modi-1 program, which one of the beauties of Modi is its ability to target multiple cancer indications. So we'll be taking this product into the clinic in the first half of next year, targeting 3 cancer indications. And then Modi-2 is in preclinical validation. And then we have a collaboration with BioNTech. Many of you may be familiar with BioNTech. It's a German based biotechnology company, mainly focused on immuno-oncology, just recently listed on the NASDAQ. And we have a collaboration with them to identify the T-cell receptors that we incorporate, so against the epitopes that we incorporate into our lead Modi-1 vaccine. So this is obviously now taking those targets into an adoptive cell therapy modality. And then we have some early-stage monoclonal antibodies that we licensed from the University of Nottingham in the middle of last year, and we'll talk a little bit about those assets too. So let's talk a little bit about our cancer vaccine platform technologies. Many of you may be familiar that cancer vaccines have not had a great track record in the clinic in previous technologies, and this is primarily due to these technologies falling short of invoking potent or high-frequency T-cell responses. And what Scancell has done is come up with 2 technology platforms that overcome those limitations. So the first is Immunobody. This is a DNA-based vaccine technology, and this actually has a dual mode of action that's very efficient in inducing very potent CD8 T-cell responses. And I'll talk a little bit about that mechanism. And then Moditope is a whole new class of cancer vaccine where we're actually now targeting stress-induced post-translational modifications. And that has a unique immune response by invoking cytotoxic CD4 T cells. And I'll again explain a little bit about that platform shortly. And of course, what we're trying to do with these technologies is to identify 4 key attributes that we're looking to look at these as commercially successful. First of all, obviously safety. Secondly, to address the unmet needs in the treatment of hard to treat, and particularly in the hard to treat cancers. but also to provide an increased and durable response. So we're invoking an immunological memory, if you like, with these cancer vaccine technologies. And then, of course, be able to have cost-effective therapies, and particularly if we're looking at combinations, we're not putting too much burden on the healthcare system. So Immunobody, as I mentioned, this is a DNA vaccine. So this DNA is delivered either using electroporation, or we can actually now have evaluated a nanovesicle delivery, where it transfects the muscle cells and also the APCs at the site of injection. And that actually has two modes of action, so we can get this efficient processing of T-cell epitopes that are integrated into this antibody-like molecule. So the DNA plasmid transcribes to this antibody protein, and in this protein we have integrated T-cell epitopes against cancer antigens. And because of this unique modality, we can actually see the efficient processing of these T-cell epitopes by antigen-presenting cells, either through direct presentation or cross-presentation, because we have a functional Fc on this molecule that allows that protein to be internalized by antigen-presenting cells. And this induces very potent and high-frequency CD8 T-cell responses. So in case of SCIB1, we've integrated T-cell epitopes against TRP2 and GP100. These are recognized cancer antigens in melanoma. As I mentioned, that product now is in phase 2 clinical study. It's delivered using electroporation. And then in the collaboration with CRUK, we're targeting multiple epitopes to NY-ESO-1. And that product will be delivered utilizing this new technology that we've identified, which will be a nanovesicle delivery. So some of the clinical data from our phase 1/2 study, you can see this is a patient with lung metastasis. We see the resolution of this lung lesion as a monotherapy in this study. But interesting data here, this is a patient cohort that had resected disease. So the little red dots there, each of these horizontal lines represent a patient, and those red dots represent surgery, where these patients have presented with a lesion, had that resected, then present again with another lesion or at the same site or another site, and obviously they still have melanoma. So at each resection, there's no measurable disease, but these patients keep presenting. And of course, they're advanced melanoma, stage 3, stage 4. So the vertical black line is steady entry, and you can see that the majority of these patients over a period of 5 years post-immunization were disease-free and without any other therapy other than the treatment with our cancer vaccine SCIB1. So that's a compelling result. And our strategy now is to look at the combination of this product with a PD-1 inhibitor, Keytruda, in a patient population with metastatic disease, which is obviously a much easier study in terms of actually looking at therapeutic response. So the reason behind that is if you look at this preclinical model we have here, if you look at the survival curve, both with SCIB1 and a PD-1, you'll see it's very, very similar as monotherapy. So we're getting around about a 60% survival. However, the orange line, where we combine these two products, we get a significant synergistic mode of action between these two, and we get a much more increased survival rate. And of course, now our objective is to see if we can translate that to clinical data. And that will be the phase 2 clinical study, as I mentioned, where we initiated in August of this year. We're just in the process of recruiting patients and expanding the number of clinical sites for that, not just here in the UK, but also potentially to the US and other jurisdictions. This will be a very relatively short study. It's about 25 patients where we're looking at the combination of SCIB1 with a checkpoint inhibitor, where the response rate in this patient cohort, this is patients with stage 3, stage 4 melanoma, inoperable disease. We generally see a response rate to Keytruda of around 30%, and we're hoping to enhance that to around 55% in this study. This will be an open label study, so we expect to see some of the interim data come out of the study as we step through 2020. Now, just like to shift track to our Moditope platform. Moditope, as I mentioned, targeting stress-induced post-translational modifications. You can imagine the metabolism in a cancer cell is very different to that of a normal cell. And so this is a rapidly dividing cell environment, hypoxic, nutrient-deprived, and that invokes autophagy. This is the recycling of cellular proteins to enhance cell survival. And what happens during that process is that you get post-translational modifications as these proteins are broken down. In one case, we see the transformation of arginine to citrulline through the PAD enzymes, upregulation of PAD enzymes, and then we also see the conversion of lysine to homocitrulline. And the inventive step that Scancell took was to actually characterize and identify these peptide fragments and recognize that these can be highly immunogenic if they can be induced to be expressed in MHC class II to induce T-cell responses. And that's exactly what we saw when we actually tested these in the laboratory. So we identified two proteins that are highly upregulated in cancer, vimentin and NLase, that are preferred substrates for these PAD enzymes. And we've identified where these actually citrulated fragments may be in terms of the immunogenic profile. And if you see from this mouse model, the sort of very aggressive B16 mouse model, where you see that all the mice are dead after about 23, 24 days. And then just immunization, either with just the vimentin or the NLA citrulated peptides, we see that we can get survival of around 80 to 90%. And if we combine those two, which is the blue line, we see 100% survival. That's an unprecedented result in this model. We've repeated this in other cancer models. We've let the tumor grow. We've seen tumor shrinkage. We've actually also re-challenged the surviving mice with tumor without any further immunization, and again seen significant survival beyond that 50-day period. So again, our objective now is to translate that to clinical data, and we're now advancing this product into the clinic in the first half of next year, where we're targeting 3 cancer indications: head and neck cancer, triple-negative breast cancer, and ovarian cancer, and will be individual cohorts. So this is not really just one study, it's about 3 studies in parallel, obviously looking for clinical responses in each of those cohorts. We actually link these peptides to an adjuvant. This is an adjuvant from isopharmaceutical called Amplavant. And what we do is combine each of those peptide adjuvant combinations into this vaccine. So in the case of Modi-1, there'll be 3 peptide conjugates incorporated, 2 from vimentin and 1 from NLase. Now I'd just like to switch track to our monoclonal antibodies. These are targeting tumor-associated glycans. Glycans are very interesting therapeutic targets, but very difficult to raise antibodies against, and this is something that Scancell has been very successful in doing. So glycoproteins and glycolipids are highly involved in cell-cell matrix interactions, but also in cell-tumor microenvironment, or modulation of the cell-tumor microenvironment. And they've been associated with the modulation of many of the hallmarks of cancer, for example, in cell proliferation, the metastasis, and also drug resistance. And so there have been interesting targets, and we've have some very interesting preclinical data where we've been able to demonstrate that antibodies directed to some of these sugar signatures we see in cancer are able to actually invoke an immunogenic cell death, so they're direct cell-killing antibodies. They also induce ADCC, CDC, and can also internalize. So they're very attractive targets for antibody-drug conjugates. And we have a body of preclinical data for each of these antibodies. While we were developing these antibodies, we also saw that when we humanized some of these are all generated in mice, when we humanized them, we actually lost some of that direct cell-killing capacity. And we were able to screen the FC between the human and the mouse antibody and identify specific residues that, when incorporated into the humanized antibody, we could actually reinstate that direct cell-killing capacity. And this is allowing these non-covalent interactions between the FC that allow greater occupancy at the site, at the antigen site. And that, as I mentioned, was able to bring back that immunogenic cell death property. And we believe that this is a technology platform that could be applied to any monoclonal antibody, not just the anti-tumor glycan antibodies that Scancell has developed. This is just a summary of the panel of antibodies in terms of some of their properties and the tumor targets. You can see there's a bit of overlap there in terms of the cancers that these glycans seem to manifest themselves, particularly in colorectal, gastric, pancreatic, where in the case of FL133, that's specific to small cell lung cancer. The interesting one that is the exception to the other 4 antibodies is this FG2811. This is an agonist antibody targeting SSEA, and this actually stimulates T-cell stem cells. So it's not a cell-killing antibody. It actually is a very useful tool in actually screening and identifying T-cell stem cells, but also induces their proliferation. So this could have some therapeutic potential in a very different way from a typical direct cell-killing antibody. These are, as I mentioned, are preclinical assets. We're looking towards transacting these at an earlier stage of their development as compared to our immunobody and moditope assets, which we obviously are taking into the clinic. So highlights for this year have been primarily driving those products towards the clinic and expanding our IP portfolio, also our clinical advisory board and some of the additional resources in Scancell, and also bringing in investment from Vulpes Life Science Fund, a little under £4 million in June of this year. They're our largest shareholder now. Martin Diggle, who's in the audience here, I'm sure will be happy to talk to you about why they saw Scancell as a very attractive investment opportunity. Martin is now on our board of directors. Going forward, our news flow will be primarily directed towards the advancement of our SCIB1 and MODI1 products in the clinic and getting the interim data and safety data from those clinical studies. And as I'm out of time, just very simply, we have 3 platform technologies, multiple products targeting multiple cancer indications, primarily targeting clinical data now, expanding our pipeline through utility of our platform technologies, looking at technology partnerships to de-risk those developments, clinical partnerships with CRUK and our clinical advisory board to enhance the clinical development of these products, and of course, engaging with industry partners who have a strong immuno-oncology franchise. Thank you.